Today we're going to talk about a concept called virulence of viruses. Basically how severe the infection is. And one of the ways we study virus virulence is by using animal models, because for most viruses we can't infect people. There are a few that are mild enough that we may have mentioned at some point here, but for most of the other viruses we have to find an animal that we can use and infect it. And typically what we do is we can take human viruses and get them to replicate in animals. And some ways of doing that are shown here. You can, for example, put a receptor for a virus into a mouse, and that's enough to make the mouse get infected. Sometimes you can put the whole viral genome into the mouse, and it will simulate a virus that's always around, like hepatitis B virus. And sometimes you can put individual viral genes in, and that's enough to duplicate the pathogenesis of infection. All right, so this is taking an animal, mouse is shown here, but it could be any of a number of convenient animals, and you put a human virus into it and you study the infection. Alternatively, sometimes that doesn't work. You can't take a human virus and get it to replicate in an animal. An example is HIV. It's been very difficult to infect animals with HIV. Only recently has it been possible, but for many years it wasn't. So in that case, what you do is you look for a naturally occurring virus of animals that mimics the human disease. So for HIV, people have studied for years simian immunodeficiency viruses, or SIVs, because in non-human primates, they mimic HIV infection. So those are the two general approaches we take to study virulence in animals. And what you would like to do is modify the viruses to understand what makes them more or less pathogenic. Now, you have to always keep in mind the saying at the top here, which is an old saying in biology: mice lie and monkeys exaggerate. What that means is that no animal model is perfect. No animal model mimics exactly what goes on in people. And no animal model is predictive in any way of what a virus will do in people. And so, for example, when pharmaceutical companies make vaccines or antivirals, they test them in a number of animal models. The FDA would like you to have 2 different animal models to test your products in. But then they're subjected to clinical trials in people. We don't just put them on the market after being tested in animals. You couldn't do that, because quite often what happens in the animal is not duplicated in people. The drug doesn't work or the vaccine doesn't work, so you have to test your drugs in people. For the same reasoning, when we do experiments in animals, it does not mean that the same outcome will be seen in people. And we do many experiments in animals that we cannot duplicate in people. And so this causes a lot of consternation for people who don't understand this. And I'm thinking in particular of non-scientists. Not too long ago, a number of investigators were studying avian influenza virus in a ferret model for transmission. And they made a virus that could be transmitted through the air among ferrets. And this caused a lot of anxiety in the world, because this particular avian influenza virus, H5N1, is very lethal when it infects people. So they assumed because this would be transmitted among ferrets, that it would also be transmitted among people. And then there was an outcry that this experiment should never have been done. But the fact is that we have no idea if those viruses would be equally transmissible among people. Everyone assumes that what happens in an animal will also happen in people. But I want you to leave this course realizing that this is absolutely not true. You should never make this assumption, okay? Mice lie and monkeys exaggerate. So here is an example of an animal model for a human virus that we made in our lab many, many years ago. And this is a model for poliovirus. Here's poliovirus up at the top. an icosahedral RNA-containing virus. And in my laboratory, one of my students identified the receptor for the virus, called the poliovirus receptor. It's shown there. We talked a little bit about it earlier in this course. And it had been known for many years that mice are not susceptible to poliovirus. Now remember what the word means. Susceptible means they don't have a receptor. Permissive means the virus can replicate in cells. Mice are permissive for poliovirus, but they're not susceptible. So another one of my students took the gene encoding this receptor and made a transgenic mouse with that gene. And those mice now can be infected with poliovirus. We have overcome the susceptibility barrier by introducing the human receptor gene. So mice normally, if you put tons and tons of polio in them, they don't do anything. The virus does not replicate. A transgenic mouse producing the human receptor, when polio is introduced into these animals, they get paralyzed, and the disease is very much like what happens in people. So this is an animal model for studying the virus infection. Many laboratories have used this to study polio, and it's an example of modifying an animal so the human virus will infect them. That said, it is not predictive of what happens in people. If you find things that you think are important, you somehow have to find a way to test them in people. So today we're going to talk about virulence and how we study it, and one of the ways that we do is in animals, as we've just talked about. Virulence is the ability or capacity of a virus to cause disease in a host, and it usually we're talking about in animals. We don't usually talk about virulent viruses in cell culture. We're referring to a host. And we talk about virulent viruses, which cause disease, and we talk about attenuated viruses, which do not cause disease. And typically, our vaccines, our infectious viral vaccines, we call attenuated because they do not cause disease. Now, that is not a very exact definition. Virulent and attenuated. So we can measure virulence and put a number on it. And there are many different ways that we can measure virulence. And these are just a few of them here to give you an example. You could infect an animal and see how long it takes for the animal to die, a very crude approximation, but it can be compared. Mean time to death. You could look for symptoms. If there are some symptoms that you can measure, fever, weight loss, for example, are common ones. Some kind of cell that's produced. Anything that you can measure specifically and precisely in the lab, you can use as a measure of virulence. Or you could measure the actual effect of the virus on the animal, the production of lesions in a specific organ. So poliovirus not only produces paralysis, which you can measure. You can say how many days it took for the animal to become paralyzed, how many limbs were paralyzed. You can also look at the spinal cord and brain pathologically and ask, are there lesions? You can look at it under a microscope. For HIV, for example, a common measure is the concentration of CD4 positive lymphocytes in the blood, because we know the virus replicates in them and destroys them. So the number per microliter of blood is a measure for how virulent the strain is. So those are just some. There are many others, but the point is you have to put a number on the virulence. You can't just say virulent and not virulent, because that's too broad a range. So here are two assays for viral virulence, just to give you a sense for how this works. On the left are mice infected with two different strains of poliovirus. And we're looking at the number of surviving animals with time post-infection. So here we've infected 5 mice with 2 different strains, and you can see that the strain labeled type 2 in red, by day 11 or so, all the animals are dead. Whereas the blue strain, type 1, all the animals are living. So here we can compare the time to death, or just total death if you wish, and compare the 2 strains. We can put a number on it, and we can say that type 1 strain is clearly avirulent, while the type 2 strain is. On the right is a different assay. On the right, we're looking at lesions in the central nervous system. We take tissues, the brain, the spinal cord, And we make sections, we look at them under a microscope, and a trained pathologist can put a number on the lesions. And so here we have different regions of the central nervous system, cerebellum, brainstem, and spinal cord. And these are animals that have been infected with 5 different viruses, as you can see labeled here. And then on the left is the relative neurovirulence score. So this is some kind of number that the pathologist assigns to the severity of the lesions. And you can see these viruses differ in the neurovirulence. So Japanese encephalitis virus in this particular animal model is quite virulent. You see high lesion scores in all parts of the CNS, whereas dengue virus causes very low neurovirulence scores. So you can compare the neurovirulence of these in this animal model by looking at lesions. One thing you need to remember, though, is virulence is a relative property. It's not an absolute property. It is influenced by many things, not just the animal that you're infecting, not just mice or ferrets or guinea pigs or rabbits or whatever the animal may be, by how much virus you put in, by the route of infection, the species we've already talked about, the age of the animal makes a big difference, the gender, and its susceptibility. Different strains of animals can vary in their susceptibility. And these are variables that we can only control in the laboratory. We can't do this, even for viruses that we can use to infect people, like common cold viruses, which cause very mild illness. We can infect people experimentally. We can't control for many variables. So you cannot compare the virulence of different viruses. We can only say that we can compare similar viruses, say viruses within the same genus of a family, and we have to assay them in the exact way. We have to use the same host, inoculate in the same way with the same amount of virus, preferably at the same time of day and so forth. And for people, we just usually cannot do that. So in a natural infection, for example, you can't say that Ebola virus is more virulent than polio because, first of all, they're different viruses and they've been inoculated into their hosts in different ways. We don't know how much virus has been received, and we don't know the genetic variability among the different people. People are not identical genetically. We're all quite different, and that influences susceptibility as well. Yes?
So when you say similar viruses, what do you mean?
So within a family, there can be different genera of viruses, right? So the family that poliovirus is in, there's the picornavirus family, and then the genera are enteroviruses, say, and hepatoviruses. Within the genus is as far as you can go in comparing virulence. You can't go even within the family. And so this is really important, and this is one of the main reasons why many people make incorrect assumptions about virulence, because it's only something that you can compare if you've set up a very quantitative assay. So here's an example of how the route of inoculation influences disease. This is infection of mice with a virus called lymphocytic choriomeningitis virus, which is a naturally occurring rodent virus. Infects mice and other rodents in nature. Occasionally can infect people who have rodents as pets. And here, if you infect by the intraperitoneal route, there's a typo there, IP, you inject the virus into the belly of the animal. You can put up to 100,000 plaque-forming units of this virus, and the mice survive. They mount a very good immune response, and the infection is cleared. But if you put one plaque-forming unit intracranially, you put a needle directly into the brain of the animal, you can put one PFU in, all the animals die. So this is an example of the route of inoculation markedly influencing virulence, because clearly you would say this virus is quite virulent here if you put it IC, but not if you put it intraperitoneally. Yet it's the same virus, right? So here is a great example of how you have to define how you're measuring virulence in order to compare even the same virus in different experiments. All right, our first question, which statement about viral virulence is wrong? One, it can be influenced by dose, route of infection, species, age, gender, susceptibility of the host. It can be quantitated by measurement of fever. Ebola virus is more virulent than human papillomavirus. It is the capacity of a virus to cause disease in a host. When comparing virulence, the assays must be the same. 94% of you answered C. Ebola virus is more virulent than human papillomavirus. Now, you may think that's true, but you can't say it because the people who get Ebola virus are inoculated very differently from the people who get human papillomaviruses. Different amounts of virus, different routes of inoculation. There are different populations of people, so you just can't compare it. And if someone died of a papilloma infection, they might disagree with you if you said Ebola virus was more virulent. So the answer is C, and the 5% of you may be quantitated by measurement of fever. Sure, you can measure virulence by measuring how much fever is induced by a virus infection. That happens quite often, or that is done quite often. So in virology, one of the major goals of our work is to identify the determinants of virulence. We want to know the genes that are involved, both in the virus and the host. So just like the viral and host genes of the immune response combine to make pathogenesis, to make the disease occur, the virulence of a virus is determined by both viral genes and host genes. And I'm going to go through a number of examples to tell you about that and how it works. We usually identify these genes in the virus by mutation. We take a virus, we make mutations throughout its genome, and then we put the viruses in animals and we ask which mutations knock out or reduce virulence. We use a very precise assay, of course, and then we try and figure out what those genes are doing. Up until not too long ago, we couldn't make mutations in animal hosts. And so it was very difficult to understand the genes in the host that influence virulence. There were some naturally occurring variants, like mouse strains, for example, that are different and have different outcomes after infection. So those were used. But now we can make mutations. We can knock out genes in mice very readily by a variety of technologies. So we are now getting a really good indication on what host genes are involved. And to make it even more exciting, in people, we can't knock out genes in people, but we can, but we're not allowed to. We can use whole genome sequencing and compare people who get very sick from a virus and people who don't get sick, because we can sequence someone's genome in a day now, right? So you can easily get sequence and ask what genes are different. Now, this is not a trivial exercise because we have 3 billion base pairs in our genome. So just analyzing the data is quite a chore. But it's now possible, and I'll show you examples of this in a bit, of identifying mutations that correlate with susceptibility to virus infection. So here's an example of how we would identify a viral virulence gene. We take a virus that grows nicely in cell culture. It makes plaques, so we can assay it. And we infect the mice with it, and the virus replicates and it causes paralysis. So this virus is neurovirulent. We then have made a mutation in this virus which affects the replication in cell culture. It doesn't grow well. You can see there are fewer plaques. We put this virus in an animal, and of course it's attenuated. It's reduced virulence because it doesn't grow very well. So this is not an interesting result. We don't want to identify genes that affect general replication because obviously they're going to be less virulent. They can't replicate as well, and they're overcome by the immune response. What you want are viruses like those shown at the bottom here. You make a mutation that does not affect viral growth. Now, there's an error in this figure. These plaques should not be smaller. They should be the same size and the same number as the wild-type virus. So this virus has a mutation in a gene that doesn't affect replication in cells. And when you inoculate this virus into animals, in the animals say it doesn't replicate well and it's attenuated. Those are the genes that we're interested in because they are specifically required for growing or some aspect of pathogenesis in the animal that affects virulence. These are what we call virulence genes. And these have been studied for a number of viruses, and they occur in different classes. So a lot of genes that have that phenotype, no effect in cell culture, but a major effect on virulence in an animal. They can affect viral replication in the animal for many different reasons. They can infect invasiveness. So the experiment with LCMV I showed you, where you put virus peripherally and it can't invade the CNS, which is why it's not virulent, that's an example of altered invasiveness. You could make a mutant virus that peripherally would not be able to get into the CNS, and that would be why it was not virulent. You can make alterations in genes that affect the tropism, the ability of a virus to replicate in a particular organ. The fourth one is a big one, modifying the host defense. We've talked a little bit about viral genes that affect almost every step of the immune response, whether it be intrinsic, innate, or adaptive. Mutations in these genes have a very big effect on virulence, because if the virus can't overcome immune responses, it's likely to be eliminated and therefore not be virulent. Genes that enable the virus to spread in the host from one place to another are also important for virulence, and genes that regulate the killing of a virus for cells. So genes involved in apoptosis, for example, or other forms of cell death, those can be virulence factors. So here's an example of a virulence gene, which is actually not a gene because it doesn't code for a protein. So many virulence genes, of course, do encode proteins, but I want you to understand that non-coding regions in viral genomes can also be virulence factors. So here on the bottom is the genome of poliovirus. It is a plus-stranded RNA. And at the 5' end, there's a non-coding region of 740-some nucleotides. The vaccine strains of poliovirus that used to be used in this country and are now used in much of the rest of the world for the eradication effort, what we call the Sabin vaccine strains of poliovirus, these are infectious viruses that are ingested and they replicate in your intestine, but they don't cause disease. So these are attenuated virus mutants. And there are 3 different strains of the vaccine strains of poliovirus, and each of them has a mutation in this 5' non-coding region that reduces the virulence of the virus. And here's the 5' non-coding region expanded, and you may remember that this is an internal ribosome entry site, an iris, that's involved in translation without a cap. The mutations in the vaccine strains are all in this stem loop structure number 5 here. This is a highly structured area of RNA secondary structure. Stem loop 5, in particular, this bulge, which is expanded at the right, has these mutations. Here are the mutations in the type 1, the type 2, and the type 3 vaccine strains. So these single base changes attenuate the virus's virulence. All right, so it's in a non-coding region. There's no protein encoded here. Nevertheless, these regions have important functions in the replication of the virus, and altering them can alter virulence. So let me show you an experiment. This is actually done in our lab a number of years ago, where we infect poliovirus receptor transgenic mice, so they can develop paralysis after infection, with 2 different strains of poliovirus. These viruses are identical in their sequence, except at one base in the 5' non-coding region, 472, which happens to be one of the sequences in the vaccine strains that has changed. So the vaccine strain has a U at this position, and the wild-type virus from which the vaccine strain was derived has a C. So one way you can measure the virulence of this virus is by infecting mice. In this assay, the virus is injected directly into the brain, and then you see if paralysis occurs. And then you calculate the amount of virus needed to paralyze 50% of the animals. And so for the wild-type virus with a C, about 9,000 plaque-forming units injected into a mouse brain is enough to paralyze 50% of the animal. So that's a number we've placed on its virulence. If you change the C to a U, again, the virus is identical except for the one base. Now you can never put enough virus in to kill or paralyze any of the animals. So the 50% dose is greater than 2 × 10⁷. No animals ever get paralyzed with as much virus as you can infect. And it's a one-base change. So this is an example of a change in a non-coding region that affects virulence. This change also affects replication. So here's another way you can measure the virulence by replication in the brain. So we're looking at the amount of virus per gram of brain with time after infection. And you can see viruses with a C replicate after inoculation, and that correlates with their paralysis-inducing potential. And viruses with a U are cleared from the brain. They do not replicate. There are also many examples of virulence genes that are involved in the modification of host defenses. And as I've said, we've talked a lot about that in this course. We've talked about modulators of apoptosis, autophagy, other antiviral proteins like ApoBec3G, virokines and viroceptors. These are viral proteins that either mimic a cytokine, the virokine, they bind to a cytokine receptor and block its action, or they're receptors. They're soluble receptors for cytokines. We call those viroceptors, and they bind the actual cytokine and block their effects. And as you may remember, the cytokines and chemokines are important for elaboration of the host response, and if a virus interferes with them, it will be more virulent. Many antagonists antagonize complement, and these are virulence factors as well. The modifiers of the major histocompatibility pathway that prevent the display of viral antigens on the surface and therefore would prevent the cell from being killed, these are virulence genes as well. So you can make mutations in these genes. The viruses will be less virulent. But those viruses do not have any defect in cell culture because there's no immune response in cell culture. You don't need these genes for replication in a cell culture, but you do in an animal, and that's why knocking them out affects the virulence. So here's an example of one of these genes. This is a herpes virus gene that is basically a virokine. It encodes a chemokine receptor. So this protein is going to bind chemokines that are produced by the host as part of the immune response and block them from working. So in this animal model, you take the herpes virus and infect mice, and the mice die, and they also develop a cellular infiltrate in the target organ. You can make sections of the organ and look at immune cells of various sorts, macrophages, neutrophils, lymphocytes, etc., coming into the infected tissue. So on the left is the survival curve, percent survival with dose of virus, increasing dose of virus on the x-axis, survival on the left. So that's one measure of virulence. Here's the wild-type virus in blue. So that's the curve. You can see a nice measure is the 50% survival point, which would be about there, slightly more than 1 PFU. Now this virus in the green curve, we've removed the gene from the virus encoding this chemokine receptor. And now you can see that the dose response curve is altered. So the 50% survival point is way out here, greater than 100 PFU. So it takes over 100 PFU to kill half of these mice and just over 1 PFU to kill 50% of the wild-type mice. Now when you do these kinds of experiments, when you delete a gene from a virus, the way you do it is you take the viral DNA, you cut out the gene, and ligate the ends together, and then generate virus. And in that manipulation, things sometimes go wrong. So to make sure no other gene has been mutated, you usually take that virus and return the gene to it and see if that restores the wild-type phenotype. And that's what MR is. In the red, they have returned the gene to the virus, and you can see it restores the lethality. And along with the increased survival of viruses with that lacking that gene, survival of mice lacking that gene, the cellular infiltrate has changed. So if you look, for example, at neutrophils, mice infected with wild-type virus have neutrophils, a certain percentage of neutrophils in this target organ. That's shown in the blue here. If you delete the chemokine gene, you see now the neutrophil response is reduced. And the particular pattern differs according to the type of cell that we're looking at. So that's an example of a chemokine receptor gene in a virus that's influencing virulence. Other viruses have similar genes encoding immune modulators. There are very few examples, though, of proteins that are actually toxic on their own. In the bacterial world, a lot of bacterial pathogenesis is mediated by toxins that are elaborated from the bacterium. Diphtheria toxin, for example, is a good example of that. It gets into the cell and damages it in some way. However, the rotaviruses, these are double-stranded RNA containing viruses that cause gastroenteritis. We'll have more to say about them next time. These actually encode a viral enterotoxin. It's one of the viral proteins called NSP4. It's a non-structural protein that is just made in the infected cell. And these viruses cause gastroenteritis, they cause vomiting and diarrhea. And it's thought that this NSP4 is mainly accountable for those symptoms, because you can produce this protein and feed it to experimental animals, and those animals will develop gastroenteritis very much like a virus infection. So a single virus protein can duplicate the gastroenteritis. And what we think this protein does is illustrated in this picture. We think the protein essentially causes an imbalance in the ion levels in the cell. So the protein is shown in the red triangles here. And these different cells illustrate the different sorts of effects that protein might be having. These are intestinal epithelial cells throughout your intestine. Here's the apical side and the basolateral side. So for example, here on the left, the virus is infecting one of them. It's producing NSP4. And that NSP4 protein is interfering with calcium storage in the cell. It's releasing calcium, and that affects the fluid levels in the intestine. These NSP4 molecules can also interfere with the way the cells are attached to one another. The tight junctions that join this epithelial sheet and make an impermeable barrier, that's disrupted by infection. That leads to fluid loss as well. We also think that the protein can bind to cell surface receptors and influence signal transduction pathways that also affect calcium levels in the cell. And finally, there is an enteric nervous system. There's a network of nerves in your gut that eventually, of course, go to the spinal cord and brain. And it's thought that NSP4 in some way interacts with the ENS. The ENS, if disturbed or perturbed, would cause you, for example, to vomit, to have contractions in your gut, or to have fluid loss. And so we think that NSP4 might be acting in that way as well. So this is an example of a viral toxin that can produce virulence. If you make mutations in NSP4, the virus is less virulent in animal models. Another example of a cellular this is actually the first example of a cellular protein that affects virulence. It's a cellular protein called TRIM5 alpha. And this explains why old world monkeys are not infected by HIV-1. They have this protein in their cells. When viruses enter, these are retroviruses that have to enter and reverse transcribe their RNA genome. There is a block before reverse transcription. And it's thought that TRIM5 somehow targets the viral nucleocapsid for degradation. So the virus in HIV infects a monkey cell, TRIM5 binds to it and in some way targets it for degradation so that reverse transcription doesn't occur. So the monkeys are resistant to infection at an intracellular block imposed by TRIM5. If you could alter TRIM5 in monkeys, you would reduce the virulence of the virus. There are also interesting non-coding cellular nucleic acids that impact virulence as well. And the one shown here is a microRNA. We've talked about these very briefly in terms of translation regulation. These are small regulatory RNAs that we produce that regulate our gene expression. One of them, miR-122, happens to be a liver-specific microRNA, which has a normal function. It is involved in regulating cholesterol metabolism. It turns out that hepatitis C virus requires miR-122 to replicate. And here's the genome of hepatitis C virus. The very 5' end is an internal ribosome entry site, an iris. And here's miR-122 in blue. miR-122 binds to the very 5' end of hepatitis C viral RNA. And it appears to stabilize it and to enhance its replication. So this is a cellular microRNA. It's liver specific. So this explains why Hep C, hepatitis C virus, is hepatotropic. A therapy is being developed based on this requirement. Because if you take away miR-122, the virus is no longer virulent. It can't replicate in liver cells. And so what's been designed is what's called an antagomer. This is a drug. It's shown here in black and red. It's called miravirsin, which will bind to miR-122 and make it unavailable for Hep C. And in chimpanzees, this drug will prevent hepatitis C virus infection, and it's now in phase 2 trials in humans and seems to be quite effective at preventing Hep C infection. So again, this is a cellular nucleic acid that is required for the liver-specific replication of this virus. Another determinant of virulence, of course, is the ability of viruses to kill cells directly. We've talked a little bit about how viruses can induce cytopathic effects by apoptosis. There are other forms of programmed cell death besides apoptosis. They're called necrosis and pyroptosis. But genes that regulate this in virus infections will determine the virulence of the virus. There are also viral proteins called viroporins that have the ability to make channels or pores in membranes. And so these poke holes basically in various membranes of the cell. The contents will then, of course, leak out and the cells will lyse. So there are also determinants of virulence as well. Many viruses inhibit macromolecular processes in the cell, protein synthesis, RNA synthesis, DNA synthesis, and this leads to an overall compromise in the structure of cells. Lysosomes, for example, the membranes around them become compromised, they leak their contents, and this basically digests the cell and causes it to die. So any viral proteins involved in these processes can be determinants of virulence. And finally, envelope viruses form syncytia. This is something we talked about a while ago at the very beginning, the ability of neighboring cells to fuse with each other because the viral glycoproteins are expressed on the surface and these give rise to fusion. That kills the cell. So again, the genes of the virus that are involved in syncytium formation are determinants of virulence. And finally, as maybe you would guess, our microbiome can regulate virulence. So we have bacteria all over us and within us. Trillions of bacteria, more bacterial cells than we have of our own. And for many viruses, the viruses have evolved to benefit from this microbiome. So here's an experiment using poliovirus transgenic mice, which was done by a virologist in Texas. And it shows that poliovirus replication in the mouse requires bacteria in the gut. So if you take mice, their guts are full of bacteria, just like people. So this is the colony-forming units per milligram of mouse feces. That's how many bacteria there are present. If you treat these mice with antibiotics, you greatly reduce the amount of bacteria in the gut. So they now took these two different kinds of mice, untreated or antibiotics treated, and then infected them with poliovirus. So they put virus through the mouth and then assayed its replication. So here on the right is that experiment. Viral replication, you just take mice feces and you measure the amount of virus in them and you look at time after infection. So the untreated animals are the solid circles. So you can see at about 24 hours after inoculation, you find virus in the feces. If you inoculate the antibiotic-treated mice, those are the black squares, you don't see any virus in the feces. The virus actually requires gut bacteria in order to replicate. So in a way, so this of course prevents disease. So the bacteria are a virulence factor because they prevent the virus from replicating. Why exactly the virus requires them isn't known, but it probably is a component of bacterial cell walls that is stabilizing viral infectivity. So the public is obsessed with viral virulence. And if you look at newspapers or online reports about viruses, you'll see a fascination with the idea of viruses becoming more virulent, either in nature or as a result of experimentation in the laboratory. And a question that arose a number of years ago, over 10 years ago now, is should we or should scientists be allowed to make viruses more virulent? So I've told you so far about how we identify virulence genes by knocking them out and making the virus less virulent, making them attenuated. But what about the opposite approach? If you make an attenuated virus, a naturally attenuated virus, more virulent, you would learn from that as well. But the public doesn't like that. They think it's scary because they think these viruses are going to get out and wipe out the world. Okay? So there's quite a bit of debate over this issue. And it really started a number of years ago with this experiment. So this is a pox virus. It happens to be mouse pox. And in Australia, they were trying to use viruses to control their rodent problem. They had a lot of mice running around. They wanted to get rid of them. And they had a brilliant idea of immunizing the mice against an egg protein. So the egg, the ova, the outer portion of the ova, they wanted to immunize the mice so that they could not get pregnant and they wouldn't have offspring. And they were going to deliver the antigen using a pox virus because it's a good vector to do that in. So it didn't work. They tried this many times. They couldn't get good antibody titers. So what they did was they put the gene for IL-4 into this pox virus. So this pox virus has a gene for an outer zona pellucida protein of the egg, and then they put an IL-4 gene into it. And the idea is IL-4 is a cytokine that biases Th2 responses, so you get better antibody production, because Th2 cytokines help make antibodies. And they said, okay, this is going to make better antibodies, so the mice will not will have good antibodies against the zona pellucida protein. Well, it turned out that putting IL-4 into this virus made it highly lethal. They had a genetically resistant line of mice to this pox virus, which is resistant because it made a great immune response. And this recombinant virus wiped them out. In addition, they had these mice that had been immunized with a wild-type pox virus, so they had antibodies against the virus. And this IL-4 producing virus wiped them out as well. So basically, this IL-4 prevented the proper expression of memory and overcame cellular responses as well. So this was published a number of years ago. It's a group in Australia, and there was an outcry over this because they said this work should never have been done. Of course, it would have been hard to predict this result, so they were doing the experiment for other reasons, but it underscored the possibility of making more virulent viruses. And so since then, it's very difficult to do that sort of experiment because this scares people. And the question really is, should we worry or not? Because we don't know. This is a mouse virus. It's not likely to infect people. There are no examples of that happening, but you don't know for sure. And this is one of the problems with science. You can never be 100% sure that something won't happen, right? We can't give you an absolute negative. We can tell you if things will happen, but not if they won't. So here's another example of this obsession with virulence. So whenever there is a virus outbreak, all right, you can do this experiment yourself. Just look at the news and how they report it. And it's always in terms of the virus getting more virulent. So if you look at the Ebola virus outbreak, after a few months, there were these news reports. Scientists think the virus is mutating and may become more virulent. And there's just this obsession with that, even though I don't think I know of any example of where that has happened, where a virus has become more virulent. There's just this idea that that's the way a virus wants to be. It wants to be more virulent so it can kill more hosts and spread, but it doesn't make any sense. We will actually talk about that in more detail when we talk about evolution. But here's an example of this obsession. This is actually an opinion paper published in Cell, Host, and Microbe by two highly respected influenza virologists at MIT. And I think this paper is a lot of nonsense, all right? There's an outbreak of influenza in India at the moment caused by an H1N1 strain. And there have been about 800 deaths. And so that's, for the number of cases, for the number of infections, it's a quite high fatality rate. Nobody knows why the virus is so virulent in this particular outbreak. But everyone points at the virus immediately and forgets that there's a host involved, and there could be a host of other factors regulating it as well. So in this paper, it's called Influenza Surveillance, 2014-15 H1N1 swine-derived influenza virus from India. The outbreak has reportedly led to 800 fatalities. The reported flu hemagglutinin sequences from India indicate that these contain amino acid changes linked to enhanced virulence. Okay? Problem is, we don't have any sequence from this outbreak. And they say this in the paper. Limited sequences are available. So what they do is they go to a 2014 isolate that was isolated before this outbreak, and they look at its sequence, and they find this change in the hemagglutinin, change in the hemagglutinin from aspartate to asparagine at position 225. And they say this has been associated with serious illness. So whenever you see the word associated, immediately your suspicions should rise. That means it's never been proven. Because in people, you can't prove that such a mutation is associated with higher virulence. So I actually tried to find the paper that describes this associated, but it's in Russian, and I couldn't read it. And it's in a journal that no one really reads, okay? So I suspect what happened is some people went to the hospital in Russia, and they were sick with flu, and they isolated a strain perhaps from one of them, and it had this change in it. And that doesn't mean anything on its own. It's just there. It's like if I tie my shoes in the morning and it rains, does that mean tying my shoes will cause it to rain? No. So just because the mutation is there means nothing. So all of a sudden it's been associated with serious illness. But then later in the paper, so they say, well, this is really bad because this sequence is in the 2014 Indian isolate. But then they say, we don't actually know if this is in the current outbreak strains. So this is a scare tactic for several reasons, and the most important one is to get funding, unfortunately. That's the way influenza works. When you scare people, you get more money because this is a virus that can spread globally. And the press has picked up on this, all right? So I got a call just before this was published from NPR, and they said, should we cover this story? Because all the other outlets are picking it up. And I told them what I told you, that this is nonsense and there's no reason to pick this up. But this happens over and over again. In every virus outbreak, there's this for some reason, an insistence on imagining things getting more virulent. Now, in the end, I'll bet this has nothing to do with the sequence of the virus. There's something about the population, the way the virus is spreading, the dose, whatever, that has caused this high mortality rate, if in fact it's true. But I'll bet it has nothing to do with the virus. But the virus is easy to blame, right? Because in fact you can get a sequence, but you cannot prove it. Next question is, which statement about determinants of viral virulence is wrong? Virulence genes can encode viral proteins. Virulence genes can encode cellular proteins. They are all the same. They are the same in all viruses. They can be found in untranslated regions. They may encode immune modulators. So 94% of you said they are the same in all viruses. You're right. That's incorrect. They are not the same in all viruses. Some of you said that B was wrong, but cellular genes can encode virulence determinants, and one of the examples I gave you of that was the TRIM5 gene regulating HIV infection of monkeys. Now, another way that viruses can be virulent, they can cause immunosuppression. And this is something we'll come back to when we talk about HIV. They can globally reduce the immune response while they're infecting. And this happens when they replicate in some cells of the immune system. They can disturb cytokine production or cytokine signaling. And, the production of viroceptors or virokines, these mimics of cytokines and viroceptors, these can cause immunosuppression as well. So here's an example of that. This is immunosuppression of a tuberculin test. Now, you may know that if you want to know if people have ever had tuberculosis very quickly, you can give them a time test in which you inject a small amount of tuberculin protein just beneath the skin, actually intradermally. And if the person has been infected with tuberculosis, you'll get a very rapid cellular response and a swelling. And you can easily see that. So here we're measuring the amount of swelling in duration during and after measles. So these are individuals who have measles, and this is the base reaction, about a 7 millimeter bump raise in the skin when you inject the tuberculin protein. And here, when someone has the active rash of measles, there's no reaction to the tuberculin. These are in known TB positive people. So you can see that measles infection is immunosuppressing. This is a mainly T-cell reaction that is occurring in the skin, and measles infection is suppressing it. And only after the rash goes away do you regain that reactivity. So nice example of measles-based immunosuppression. And the way virus does it, the virus infects dendritic cells and monocytes, and it interferes with their ability to present antigens. The amount of circulating T lymphocytes is decreased by about half. And these T lymphocytes are important for the tuberculin test, and that's why it's interfered with it. And you also get aberrant cytokine responses. You have increases in IL-4 and IL-10. And just to remind you, IL-4 and IL-10 skew the T-cell response to a Th2 type, which favors antibody responses. So measles is pushing the response towards antibodies and away from cellular responses, and that's why it interferes with the tuberculin test. Other viruses immunosuppress as well besides measles. Rubella immunosuppresses. It infects lymphoid cells. And HIV, of course, infects CD4 positive T cells. It infects helper cells. So it screws up both antibody production and cellular responses. And you get many opportunistic infections as a consequence, which normally you can deal with. So let's look at now some host genes that are involved in regulating virus disease. So these are examples of virulence determinants. Here's one figured out by studying strains of mice that differed in their response to infection with a flavivirus. And here we have two strains of mice, C3H8E. And these mice, when you infect them with the virus, they all die within a few days or so. So these animals are completely susceptible. Then on the other hand, there's another strain of C3H mice called RV, which are relatively resistant to infection. So this is the virus replication in the brain. So you can see the susceptible ones, you get a lot of virus replication accompanied by death. The resistant ones have much less virus replication, although half of them may die anyway. The control of this infection is by a cellular gene called FLV. It was originally called FLV for flavivirus because it controlled susceptibility to flavivirus. It turns out to be the gene encoding 2', 5' oligo A synthetase, which is an innate immune response gene. It, together with RNA cell, degrades RNAs and protects cells from infection. So these susceptible mice have a mutation in FLV, and that's really what makes them die after infection. Human determinants of susceptibility, as I said, are beginning to be known. A very famous one is the CCR5 gene, which is a co-receptor for HIV. Now remember back to our entry talk, HIV binds to two different receptors on the host cell, CD4 and a chemokine receptor, two different chemokine receptors. One of them is CCR5. About 4 to 16% of people of European descent have a mutation in the CCR5 gene. They don't produce the protein. Now apparently this is of no consequence because they're otherwise healthy, but they are resistant to HIV infection. And a number of years ago, you may know this story, a German patient had AIDS. He also had lymphoma, so his physician decided to give him a bone marrow transplant. This is a procedure where they irradiate your bone marrow and then replace it with that of a donor. And his physician was very smart. Not only did he get a matched donor, but he found someone who had a deletion in the CCR5 gene. And this German patient is now cured of AIDS because his cells are no longer susceptible to infection because they lack this gene for CCR5. So because of this patient, now many people elsewhere are looking at ways to take AIDS patients and disrupt the CCR5 in them to make them resistant to infection. And there have been some hints of it working so far, but it's going to be a while before this happens. Herpes simplex viruses are we'll be talking about these next week. They cause persistent infections in everyone. In particular, herpes simplex type 1, we acquire very early in life from our parents. And the virus infects our mucosal surfaces initially and then establishes a latent infection in our sensory and autonomic ganglia. So typically herpes simplex type 1 we acquire in our respiratory mucosa, and then the virus becomes latent in our ganglia up in the neighboring area. And periodically the genome is activated and virus particles are produced and you get a fever sore. Sometimes the virus goes beyond the lip and forming a fever sore and gets into the central nervous system. And this happens at about 1 in 250,000 people per year, and it's 70% fatal if untreated. And this happens at very young life, so it can be associated with a primary infection from your parent, or it can happen later in life when the virus is reactivated. And so a group here at the Rockefeller wanted to know if this low frequency of encephalitis had anything to do with genes in the host that were altered. So they did whole genome sequencing of a number of individuals who had recovered from herpes simplex encephalitis, and they found mutations in these genes, TLR3, UNC93B TRIF, or TRAF3. These are all involved in sensing herpes simplex virus DNA. TLR7, you may remember, is a component in the endosome that senses viral DNA. When it does so, it stimulates the production of interferons and other cytokines. And TRIF and TRAF are proteins that are part of the TLR7 signaling pathway. So if you take those away, you have a defect as well. And then UNC93B is a protein that helps get TLR7 from the endoplasmic reticulum to the endosome. So the mutations in these patients all affect TLR7 function. They have point mutations or deletions in these proteins, and that makes them more likely to get herpes simplex encephalitis. And in particular, you can take these mutations, you can introduce them into cells and culture, and you can show that they allow higher virus replication. So it's a very nice combination. So it's a great example of mutations in innate immune genes that predispose to serious disease. Another group of such mutations has been identified in patients with severe influenza. So again, they get patients, large numbers of patients who are hospitalized from severe influenza, which is a rare event. They sequence their genomes and they find a change in this protein, IFIT-M3, interferon-induced transmembrane protein number 3. And in cell culture, the mutations in this gene affect virus replication. The IFIT-M3 seems to be involved in blocking binding of the virus to receptors. It seems to be involved in blocking endocytosis and fusion. So this is not as tight a case as the herpes simplex virus polymorphisms, but it looks pretty good for influenza. And these proteins also seem to be involved with other virus replication as well. So these are two examples of how you can use genome-wide sequencing, or genome-wide associations, GWAS, to identify single nucleotide polymorphisms, we also call them SNPs, that track with more severe disease. And then hopefully you can do experiments in cells to prove that. Many genes that influence virus susceptibility map to the MHC protein genes. The major histocompatibility proteins, MHC1 and 2, remember they display peptides on the surface of an infected cell so that CTLs can kill them. They also are on the surfaces of dendritic cells or other antigen-presenting cells. They present them to the lymph node. So the ability to recognize peptides as foreign is very important. And there are we have many genes encoding these MHC proteins. We call them polymorphic. And typically, populations with restricted MHC polymorphism, like island populations, they have greater susceptibility to infectious disease because their MHCs can't recognize the same broad selection of peptides derived from virus infection. So there are many examples of susceptibility tracking with MHC gene. So here's an example in mice. These are two strains of mice that differ only in a MHC gene, H2AA and H2AB, and they have a very different response to virus infection. So we're looking at the enlargement of a spleen after infection with a specific virus, and you can see that the two MHC haplotypes behave very differently. And again, this is because without the right peptide binding groove in the MHC, you can't control the virus infection. This also seems to apply to human infections. We have a number of individuals infected with HIV-1 called elite controllers. They're infected, they produce low levels of virus, but they never proceed to AIDS. And Magic Johnson is an example of an elite controller. These individuals, we're trying to understand what makes them resistant to disease and so able to control viral loads. And there are many things that are at play here, but one of them is a specific MHC1 allele called HLA-B57. And we think that this particular allele is able to recognize all the different peptides that the virus produces during a long-term replication, which would otherwise escape other MHC pockets. But this one is good at recognizing all of them. So as the virus replicates, it makes variations that change the peptide sequence that's displayed in the MHC. And B57 appears to be particularly good at recognizing anything that the virus can throw at it. Viral co-infections are also determinants of virulence. And this is an interesting story, co-infections of herpes simplex virus types 2 and HIV. So the observation is that people who have simplex type 2, which is a genital infection, causes lesions in the genital area, they're more susceptible to HIV infection. And the original idea was that simply there are open lesions there, and that makes HIV more readily taken up. But that turns out not to be correct. There's actually a very specific mechanism. HSV2, simplex type 2, enhances infection of the circulating dendritic cells in the mucosa called Langerhans cells. And what it does, it stimulates the production of an antimicrobial peptide in the mucosa called LL37. And that's shown in the bottom here. Here is the mucosa infected with HSV2, and the mucosal cells are producing this antimicrobial peptide, LL37. So it's part of the normal defenses to produce this antimicrobial peptide. Well, that peptide upregulates CD4 and CCR5 on dendritic cells. It's part of its effect. It's part of what this antimicrobial peptide does, because these molecules, of course, are important for responding to various pathogens. But of course, CD4 and CCR5 are the receptors for HIV. So you more effectively can infect the Langerhans cells in the presence of HSV2 because you're upregulating the receptors and the virus is more easy to get into cells. Next question is, which of the following are examples of SNPs that control susceptibility to virus diseases? Viral co-infections, CCR5 delta 32, mutations in TLR3 that predispose to herpes simplex, encephalitis, the FLV gene, or all of the above. Look at the question again. Which of the following are examples of SNPs? You know what SNP stands for? A single nucleotide polymorphism. All right? So the answer is C, mutations in TLR3. Those are SNPs because co-infection is not a SNP. CCR5 delta 32 is a deletion in the gene. The FLV gene is also a deletion. It's not all of the above. So it's TLR3 mutations that predispose to herpes simplex encephalitis. So I want you to understand that a SNP is a single gene, a single nucleotide change that can influence the outcome. I want to close with just a few other determinants of virulence that we clearly know of. They're real, but we don't know how they work. And one of them is age. Typically the very young and the very old are the most susceptible to serious disease. We think the young because they have an immature immune response. They have an immature immune response In some cases, this can help because they don't get immunopathology, but it can also lead to more serious infections. And the older, typically for respiratory infections, the alveoli of the lung are less elastic. They're less able to breathe as easily, and you have a diminished cough reflex. And that's shown in this influenza mortality curve from 1911 to 1915. So we're looking at the death rate in different age groups. So you can see less than 5 years old and greater than 64 years old in this particular study are more highly susceptible to dying after influenza. And this is a very typical pattern for influenza. There are exceptions to this. It doesn't apply to every infection. Poliovirus and mumps and measles, for example, are actually mild at a young age. And we think this is because there's less immunopathology. These may be infections where immunopathology is a substantial part of death. 1918 influenza is an exception to this general pattern because people who were about 18 to 30 were also highly susceptible. So at the top is the outcome for 1911 to 1915. In 1918, there was a very large global pandemic with millions and millions of people killed. And again, the very young and the very old were susceptible, but there was also a peak of death, higher than a normal death, among this middle age group. And we don't know why this is so. This is actually being investigated by many people because it's interesting to understand what conferred this. And we think it may have been the lack of immunity in that particular age group, but it certainly hasn't been sorted out. Other determinants of virulence, sex. Males are slightly more susceptible to virus infections, but pregnancy is also predisposition to more serious disease, especially with these viruses. Malnutrition certainly increases susceptibility because it has a direct effect on the immune response. And for this reason, in parts of the world where nutrition is very poor, measles is 300 times more lethal than it is, say, in the US. So malnutrition is affecting immune barriers and immune function. Cigarette smoking, for sure, increases susceptibility to respiratory infections because it compromises the mucociliary elevator and it also interferes with innate immune responses. Air pollution as well, for sure, although we don't know the specific components. And stress. When you get stressed, you get virus infections. The most obvious one is a herpes simplex virus fever sore. Often people respond to stress by getting fever sores. And again, we don't know how this works, but somehow the stress is playing into the immune system. I just want to leave you with this very interesting finding that stress spelled backwards is desserts.